All right, so the talk I'm going to give today is going to be somewhat speculative, as we're just now starting to collect data as the lab gets underway. All right, so I've always been interested in how the brain makes an internal representation of our external world. And lately I've been thinking more and more about how these internal representations are a product of and influenced by our state of being. For instance, our emotions, our health, and of course, our metabolic state, which is what brings me here to talk to you all today. So with that in mind, I'm a neuroscientist and not really an endocrinologist. So I've tried to make this talk accessible to folks of a broad range of backgrounds and interests. However, if I slip into terminology or phraseology that you're unfamiliar with, please don't hesitate to chime in and give me the opportunity to rephrase or clarify myself. All right, so one of the broad questions that myself and my lab is interested in is how blends or complex mixtures of sensory stimuli are recognized, detected, and encoded by the brain. And to answer these questions, I think the olfactory system is really quite an ideal model. For example, in any room, there are dozens or even hundreds of odorants which comprise what's called an olfactory scene. And right now it's a bit early in the day, and if you happen to be enjoying a coffee as you're listening to this talk, you might want to smell that coffee above and against all the other odors in your room. So likewise, as we sit down to a meal, breakfast, lunch, or dinner, it's important that our brain can accurately interpret its olfactory composition, which ensures faithful sensory representations of nutritive substances, which we need to thrive and go about our day. So the work I'm going to talk about today describes how odor mixtures, like those found in food substances, may be represented at the interface between bottom-up detection-based processing from the nose to the brain, as well as top-down analytical processing from higher order structures in the olfactory system, including the olfactory cortex, and how context may modulate what we interpret as a sensory scene. And then finally, I plan to tell you about some of our future plans to understand how sensory processing is modulated by internal states, including energy balance. So to outline this talk, I'll first briefly talk about my research programs to introduce, what I do and what our approach is. Then I plan to tell you a little bit more about a project that's currently in progress in the lab. It's a little bit more focused on sensory neurobiology. And then lastly, I'll conclude with a project that I hope to get off the ground with a new postdoc who's going to be joining the group later this year, who you just heard from prior to this talk. So in my lab, we study neuroscience and sensory perception using a multidisciplinary approach. We observe animal behavior, and we measure electrical properties of neurons and synapses to understand how cells communicate and influence that behavior. And what I'll spend most of my talk on today is actually another approach where we study network encoding. In other words, how groups or populations of neurons respond to sensory stimuli and control behavior. And together, these approaches allow us to understand how the brain of, say, this mouse here interprets the sensory environment. In this case, an odor plume coming from this chunk of stinky cheese. So we need to understand the animal's behavior, how the neurons are connected and communicating with each other, and how populations of neurons make sense of changing conditions within our environment. So together, the experimental approaches that we use allow us to study neuroscience and sensory perception across multiple scales in the brain. At the most macro level, we study a sensory system, in our case, the olfactory system. We study networks, like those found in subdivisions or processing centers of the olfactory system. We study individual neurons. How do cells change their encoding properties as animals learn? And we can break this down even further. We can study how neurons are connected to each other and how these connectivities change based on conditions in the environment. And then finally, we study how individual molecules located within a single synapse can be adjusted to fine-tune the connectivity and network encoding properties of a sensory system. So with that in mind, my basic approach of my laboratory first aims to reveal, the neural encoding schemes used for complex sensory stimuli, including food. We hope to uncover how learning and prior experience changes sensory perception. If I've eaten something, it's made me feel ill. How do I perceive perceive that same substance in the future? And how does that contribute to consumption behaviors? And then finally, we hope to understand how stimulus encoding and learning are affected by internal states, including hunger and satiety. So although we're a sensory neurobiology laboratory, our goals and questions are also in close alignment with what we feel the programmatic goals of the National Institute of Diabetes, Digestive and Kidneys Disorders puts forth. And I want to highlight a subset of a passage which focuses on nutrition disorders and diabetes. So it says that the mission of the NIDDK is to conduct and support medical research on diabetes and other endocrine and metabolic disease, digestive diseases, nutritional disorders, and obesity, as well as kidney, urologic, and hematologic diseases to improve people's health and quality of life. So our work, I think, directly applies to these nutritional disorders and obesity. How do our internal states influence our sensory perception of food objects, and how does that contribute to consumption behavior? So our work will directly contribute to this mission by uncovering how the brain interprets nutritional content through sensory cues, and along with how that interpretation is modulated by both brain state and context. So, coming back to our model system, why do we study olfaction? And the simple answer is that olfaction plays an outsized role in the multimodal sensory construct of flavor. And it also allows us to evaluate food substances well before our other senses can contribute to that evaluation. And part of this is that our sense of smell is tasked with understanding and evaluating somewhat complex sensory environments. So in this scene here, I'm showing you a classic view of the Chicago skyline and some of our more famous buildings. And this is actually a view right outside our lab. So we've got sort of the prime real estate, as I said, looking towards the loop. And if we look at this visual scene, these aren't just buildings. They're an amalgamation of things like brick, windows, doors, and concrete that together make up a unified perception of a skyscraper, which your brain recognizes. And we can sort of frame this problem in the context of the olfactory system. Try to remember the first time that you experienced, the smell of coffee. And you just sort of think coffee as this, unified, what we consider a holistic perception. However, if you're like me, I'm just starting at my lab and, I'm drinking a lot of coffee now because there's a lot of things that need to get done. And as I'm drinking more coffee, I'm beginning to be able to recognize specific characteristics or, notes which are contained within coffee. Things like how roasted it is, how fruity it is, or perhaps how nutty it is. And these things together make up the unified holistic perception, yet they can vary on the cases of individual cups of coffee. Starbucks coffee tastes a little bit burnt to me, whereas Dunkin' coffee tastes a little bit more nutty to me. Yet I still recognize these sensory objects as coffee. So the question my lab studies, or one of the questions that my lab studies, is how does the brain take a holistic sensory representation of a stimulus and decompose it into those parts through learning? How do we deconstruct coffee into the components which together make up coffee? And more specifically, how do the stimulus coding properties of established circuits change through this learning process and brain states associated with these stimuli? If I'm thirsty, maybe coffee smells quite good, but if I'm not thirsty, maybe a cup of coffee isn't what I have in mind. And another way to think about this is how our brain interprets, this classic, fall feast that awaits us just a few weeks down the line here. As we walk into a home with Thanksgiving dinner in front of us, we're confronted with an intricate and complicated blend of olfactory stimuli. And we need to be able to answer some fundamental questions about this meal before we consume it. So things like, what nutrients are available? Are these nutrients compatible with my physiological needs? And maybe perhaps most importantly, given my past experiences, can or should I consume the available nutrients in this meal? So I want to make clear before I move on in the talk, when I say sensory mixtures, I'm talking about mixtures within a modality, not across sensory systems. In other words, how are blends of purely olfactory stimuli recognized by the brain, rather than their interactions between, say, with the visual or auditory systems. And this is an important concept because mixtures of stimuli within a given system often give rise to very different perception than the sum of those parts alone. And I think we're all aware of this phenomenon. You've undoubtedly seen this diagram of added colors, the additive color Venn diagram, where the addition of two independent colors give rise to a completely new perception. So red and green together give yellow, where it's hard to see red or green inside the perception of yellow itself. And this concept is present throughout all of our other sensory modalities. And another famous example is in our auditory system. And this is a auditory illusion, which is called combination tones. And the idea is that if you play two pure tones at the same time, the bars in white, your brain will actually perceive a third lower pitched tone being played at the same time. So our brain constructs objects based on these sensory mixtures, which aren't actually there. So I'm going to try to play this sound right now so you can get a sense of what I'm talking about. It's really kind of a fun example. And it's going to move through these different notes. And I want you to listen very carefully. And what you should hear is two higher pitched sounds and a lower pitched note being played at the same time. And that lower pitched note is not actually being played by my computer, but rather is being constructed by the neurons in your brain. All right, so I hope you're all able to hear that. And the point that I want to make is that it's very important to understand how these small components come together to make these larger sensory objects. So back to olfaction. And at this point, I want to introduce you to some of the relevant neuroanatomy as it pertains to the rest of the talk. So in this case, just to be clear, this is a mouse brain and not a human brain. And in the first part of the talk, I'm going to discuss the initial stages of the olfactory processing hierarchy, sensory input and encoding at the level of input to the olfactory bulb, and then downstream at the olfactory cortex, where odors and odor objects are recognized and categorized. In the second part of my talk, I'll talk a little bit more about olfactory perception and its potential modulation by hormones and physiological states via the hypothalamus. And I also want to highlight a secondary structure right here, which is labeled OT for the olfactory tubercle, which is going to be the focus of the future directions portions of the talk at the conclusion. And I just want to highlight that the hypothalamus provides direct neuromodulation through neural connectivity, as well as hormones through the hypothalamic pituitary axis to two very important olfactory structures, both the olfactory cortex as well as the olfactory tubercle, which is responsible for sensory reward associations. So in mammals, both rodents and humans, olfactory receptor neurons are found in the olfactory epithelium. And these are true neurons which are sensitive to a molecular feature of an odorant molecule. So here the red neurons are sensitive to the red molecule, the blue to the blue, and the green to the green. Although they can also bind to different features across a graded spectrum of affinities. So these sensory neurons of a common type, meaning those that express the same receptor protein, project an axon to a well-defined structure on the surface of a portion of the brain called the olfactory bulb. And these axons coalesce and converge at these structures called glomeruli. And in these structures, an initial round of information processing takes place, and then information is then routed towards higher order structures in the olfactory system where analytical sensory processing takes place. And here we're going to focus primarily on the olfactory or the piriform cortex. Now, importantly, this pathway or this processing hierarchy is actually bidirectional. And what I mean by that is that sensory information is sent back from the olfactory cortex into the olfactory bulb. And in fact, similar to other sensory modalities, including both the visual and the somatosensory system, these feedback projections from the highest levels in the brain actually outnumber the afferent inputs from the sensory periphery. What that means is that our cortex provides more input to the olfactory bulb than does our nose. Therefore, these projections from the cortex very likely play an important role in how we perceive sensory stimuli and provide flexible encoding schemes and on-demand modulation of sensory processing. So this is a little bit, cartoonish, and I want to zoom into what these cells, look like in a little bit more realistic form. So this schematic is a circuit diagram of the cells that we're going to be talking about in the subsequent slides. So it shows the interplay between the bottom-up peripherally driven processing from the nose and the top-down input from the olfactory cortex into the olfactory bulb. And this cartoon, again, is greatly simplified, but it does show many of the relevant cell types and the rough configurations in this circuit. So these olfactory sensory neurons convey information from the nose into the olfactory bulb via these axon terminals here, where they terminate in these olfactory glomeruli. And these glomeruli are innervated by output neurons called mitral cells, which take information from the olfactory bulb and transmit it to the olfactory cortex. Then the olfactory cortex processes that information and sends a subset of it back into the olfactory bulb, where these neurons primarily synapse with a class of inhibitory neurons called granule cells, which provide lateral inhibitory connectivity with mitral cells associated from different glomeruli, meaning different olfactory inputs. So we can sharpen or adjust the weights of the activity coming from these output mitral cells to the cortex itself. So the olfactory system is really, a great model to study the convergence of bottom-up and top-down information processing streams because of its modular and well-segregated anatomy. So to remind you from the previous slides, when odors are inhaled, they activate a subset of sensory neurons in the nose, which then projects to these structures in the olfactory bulb called glomeruli. And then each glomerulus is innervated by these output neurons called mitral cells, which project to only a single glomerulus. Therefore, stimulus specificity in the olfactory system is a labeled line, and it's maintained all the way through the level of olfactory bulb output. Now, these mitral cells then send information to the cortex, where these cortical neurons receive input from many different mitral cells and therefore glomeruli. And this is really the first point in which stimulus specificity is lost in the olfactory system. So let's consider you're enjoying that cup of coffee that's in front of you, and nuttiness is the olfactory note in coffee that you appreciate most. In this circuit, we can see that the nutty odor activates a subset of sensory neurons, which project to a single glomerulus. And then output from this glomerulus is directed down the axons of these output mitral cells. And the axons of the projection neurons head towards the cortex, where an ensemble of cortical neurons is activated. Now, of course, nuttiness doesn't stand alone in coffee, right? There's those other characteristics that perhaps we don't care about as much, things like how roasted it is or whether it's fruity or not. And these other odors activate their sensory neurons and their glomeruli, and of course, their mitral cells and their ensembles in the piriform cortex, which will, of course, overlap to some degree with the cells responsible for encoding that odorant, which we do care about. And then what we're interested in is how these neurons, which receive converging input from the on-target and the off-target odorants, send information back into the olfactory bulb to sharpen the activity of the on-target odorants. And we can think about this as well as the nutritive substances that might be in front of that Thanksgiving meal in front of us. Do we smell the short chain fatty acids, which tell us whether the meal is rancid or not? Do we smell the sugars or the protein composition? And how does our brain interpret what's in the meal in front of us? And most importantly, in the olfactory cortex, how do we deconstruct this sensory scene to understand what the composition of the food actually is? So the question that we aim to study is, does the activity of these projections from the cortex into the olfactory bulb faithfully represent multiple odors, or are they selected to only a portion of an odor mixture? And can we tune this representation based on learning and context context. All right. So to answer this question, the approach that we've begun to undertake in my lab is to use a virus to label neurons in the piriform cortex with a fluorescent indicator that increases its fluorescent profile when a cell's become active. And I'll refer to this indicator as GCAMP for the rest of the talk, and I'll talk a little bit more about exactly what GCAMP is in a subsequent slide. So here we've infected these neurons with this virus, and what I'm showing you is a sagittal section through the mouse brain. You can see the virus injection tract, and on the bottom here is the piriform cortex. We can see dense labeling of piriform neurons with this fluorescent indicator. And this is now a coronal section. I've highlighted the piriform cortex, and you can see that we can target this region with both the GCAMP as well as a second red indicator, which helps us to align the images. And this is just a zoomed in image of what these neurons actually look like in the piriform cortex. It's a 3-layered structure with the layers at the bottom doing most of the computation and the input from the olfactory bulb arriving at the top of the image in this layer called the lateral olfactory tract. So since GCAMP is going to be coming up quite a bit throughout this talk, I just want to mention that it is a modified green fluorescent protein that becomes brighter when a neuron is active. So here, these are neurons at rest, and what we can see is that they're relatively dim. But if we add some potassium to the bath that surrounds these neurons, they begin to glow brighter, and potassium increases the neural activity. So we can use this fluorescence as a proxy of overall neural activity over time. So here, if we track the fluorescence of these cells, and each time we apply potassium, you can see this robust and steep increase in their fluorescent output. And importantly, this fluorescence is directly proportional to the real electrical output of a neuron. So here, a single neuron is being recorded using an electrode. And what we can see in the electrical trace at the bottom is every time that this neuron fires an action potential, the fluorescent output of this neuron subsequently increases. And the GCAMP signal follows very closely. So this provides us a way to read out the activity of hundreds or thousands of neurons in real time in living brain tissues. So here I just want to show another little image of what these neurons actually look like in the piriform cortex when they're infected with the GCAMP virus. And because I haven't actually talked much about neurons beyond the hypothetical, I just want to mention that each of these cells has 3 parts: a cell body right here, the brightest portion of the neuron, a dendrite, which extends upwards and receives input from other neurons, and an axon below, which sends information back into the olfactory bulb. And it's these axons in this layer down here, which we actually intend to study rather than the cell bodies in the piriform cortex themselves. So since we're studying those axons, what do they look like in the olfactory bulb? So we've infected these neurons in the piriform cortex, and now I'm showing you a sagittal section of the olfactory bulb. And you can see the green GCAMP labeled axon projections from the piriform cortex descending and terminating at the deep layers of the olfactory bulb itself. And if we take a closer look here, we can see that these axons densely target a layer of the olfactory bulb called the granule cells, but in fact, they actually extend, all through the different layers, just at different densities. And here's a zoomed in here. So we can really infect these things quite robustly with this calcium indicator. And this provides us a way to assay the activity of the entire piriform cortex by looking at the activity of this population of axonal projections into the olfactory bulb. So before I show you how these axons or these neurons respond to odors, I want to tell you a little bit about how we actually deliver the odorants to the mice in our lab. So we construct these devices called olfactometers, and this is what one of these things look like. And typically they have to be a binary tree. So either 2, 4, or 16 odors in series. And we take the air, we split it 16 ways, and we control these solenoid valves using an Arduino or some LabVIEW code. And then we can open any one of these valves in concert to deliver a single odorant to the mouse, or any mixture containing up to 16 different parts. So we interface this device with a 2-photon microscope, which allows us to visualize neurons in the brains of living mice at the level of single cell resolution. So this is what this looks like when we combine the two. This is a mouse, and I'll show you a closer view. This is the olfactometer, and the odorants are being delivered to this mask, which sits around the mouse's nose. And he's running freely on a ball here to sort of keep him calm and keep his head in place. And we're visualizing the neurons through a craniotomy in the top of his skull. And typically what we do is this trial-based structure where we image the cells for 5 seconds. Then we provide the odor to the mouse for somewhere between 2 and 5 seconds. And then we image for another post-stimulus period of 10 to 15 seconds to collect the neural activity. So with this equipment, we can deliver a diversity of odors to the mouse and vary any number of properties about them or the type of information which they convey. Things like functional groups. Are they esters or not? Are they aromatics? Their carbon chain length. Things like short chain fatty acids, which have rotten type smells. And these odors can also be characterized by their functional responses when actually calculated from neural activity. So we can make any combination of 16 different odors and build blends which convey specific information about olfactory scenes or perhaps nutritive compositions of food substances. All right. So to measure these odor responses in live mice, we first make a craniotomy over both olfactory bulbs, where we remove the skull, and it exposes the dorsal surface. We then implant a thin piece of glass and seal it to the surrounding bone, which gives us optical access to the brain tissue below it. So we'll zoom in a little bit closer here. This is the glass implanted into the skull, and you can see both hemispheres, the left and the right, of the olfactory bulbs through this window. Along with that, we also implant this titanium head bar here, which holds the mouse in place underneath the microscope. So once we make these craniotomies, the mouse can actually live like this for months and even up to a year, which allows us to longitudinally follow the activity of the same groups of neurons across brain states and different contexts, and even through learning as the activity profiles begin to change to selectively encode different components of olfactory scenes. And then, of course, we image through this window, and we're imaging the projections from the piriform cortex into the olfactory bulb. And this is the typical field of view, what we might see. You can sort of see this, what looks like sparse expression of these axons and their swellings, which are individual synapses in the olfactory bulb. And I'll play this movie, and you can see that we're moving from dorsal now towards ventral. And as we move more dorsal or more ventral into the brain, we can start to see more of these axons and their synapses begin to pop up. And of course, if we can press this, we can see that we can now identify many hundreds or even thousands of synapses or axons within the same field of view. All right. So how do these axons, as well as their synapses, respond to odors? And to start our experiments, we begin by delivering odors to anesthetized mice and measuring the activity of these feedback projections. So here I'm showing you an example field of view in the mouse brain. And these squiggly lines are the axons from the piriform cortex into the olfactory bulb. And little circular points are the individual synapses that are making with their postsynaptic partners. And we'll give this odor called 2-ethylhexanol, which has kind of a fruity characteristic to it. And I'll play this movie, and you'll see that a subset of these axons and their synapses respond to this odor presentation. So you can see down here at the bottom, this cell begins to respond quite robustly to the odor. And of course, we don't just do this for one odor. We do it for many dozens of odors, things that have different smells, like citronella is kind of citrusy. And what we can identify is different subsets or fractions of these projections into the olfactory bulb, which respond to odor presentation. So this is a summary of our approach. So we image the axons in the olfactory bulb. And in the middle here is a image of what this GCamp fluorescence looks like. And from this GCamp fluorescence, we can measure the fluorescence changes over time and build what's called a spatiotemporal parallelogram. So we can look at the pixels in an image which co-vary in both space and time, which allows us to highlight areas of the image where we expect a neural response to occur. We then use an automated algorithm to, with a classifier to identify boundaries around these different subcellular compartments, which we can then extract the fluorescent signals from. So in the bottom here are some schematized images of how these axons change their fluorescent profiles in response to a given odor. So the data here is z-scored, and this is a measure of how much variation from the, or how much deviation from the baseline activity there is when the odor is presented. And what we see is that many of these axons increase their fluorescence or activity levels, and many of them decrease their activity levels. And we do this across different odors, and what we can see is that different subsets or different populations of axons respond to these different odors. So we next wanted to understand, as a first step in this experiment, the odor tuning of the feedback projections in the olfactory cortex and its relationship to the feed-forward inputs from the nose based on their spatial locations. So here we image the axon terminals of these projections from the piriform cortex into the olfactory bulb. And then for each of the synapses or axons, we measured how many of the 16 odors in our panel elicited a significant response from that cell. And what we found was that from the feedback in the piriform cortex, each of these neurons was responsive to around 4.5 odors out of the 16. Now, as a point of comparison, we did the same experiment, but measuring the activity of neurons in the nose. And I'm not going to have time to talk about how we did this, but in brief, we removed the bone over the nose of the mouse, and we could put the objective into the olfactory epithelium and image the activity of individual sensory neurons as the mouse was smelling an odor. And from these neurons, we could then measure how effective our panel of odorants was at eliciting activity in these sensory neurons. And what we found is that when we compare the feedback tuning to the feed-forward input to the nose, the neurons in the nose are more narrowly tuned, meaning that they respond to only 3 out of the 16 odors rather than 4.5. So what this tells us is that, what we have is it's somewhat unsurprising. We expect, because of this convergence of inputs from the nose to the cortex, that the cortical neurons are going to be more broadly tuned. But what we wanted to know is, what is the relationship between the neural encoding patterns between these different odorant molecules, which share some of the same characteristics? And what we use is a measure called representational similarity. And this is a standard measure used in neuroscience to understand how different stimuli are encoded by the same population of cells. So what we can do from the 16 odors is we can measure the response from any given population of cells. So this would be the relationship of odor 1 to odor 2. And we can measure the response of neuron 1, 2, 3, all the way up to neuron X, where the response is normalized on a graded scale from 0 to 1. And the similarity is simply going to be the correlation of these two vectors. And we can look at the Pearson correlation, we could look at the cosine similarity, which is going to be the angle between the two vectors. There's many different ways about doing it, but right now I'm going to show you a first pass using Pearson correlation coefficients. So if we look at these odors at the level of sensory neuron input from the nose to the brain, and these are all neurons which all odorants which contain an ester group or a tiglate on them. And what we see is, when we look at the population correlations in the nose, is that there is some structure between these different odorant molecules. So these are indicated by more yellow colors. However, if we take odorants that don't contain the ester groups and compare them to odors with the ester groups, we see that some of that similarity begins to break down. However, by and large, there's some patterned similarity in terms of the neural responses at the level of input from the nose to the brain. Now, when we do the same comparison, looking at the descending input from the piriform cortex back into the olfactory bulb, what we find is that this correlation or the structure is completely lost. there's no relationship between any single odor in terms of the piriform cortex encoding as there is at the level of the nose into the brain. And what we've done is then just simply look at the relationship between these correlations. And what we can see is that the piriform cortex is fundamentally reformatting how information is conveyed from the nose up to these analytical processing centers at the highest level of the olfactory system. So to summarize, so far we've shown that individual feedback from the cortex is more broadly tuned than feed-forward inputs from the olfactory system. And that at the population level, the representational similarity is greater in sensory neurons at the lowest levels of the system than it is in the piriform cortex. But the question that we're really after is, what about mixtures where cortical neurons, they must integrate information about multiple mixture components? Things like a meal where it's not just a single odor, but rather dozens or hundreds of odors being presented to you at the same time. And for our next experiment, what we did was, try to simulate just that. We made complex olfactory mixtures using the same initial 16 odors in our panel. So in this experiment, we devised 100 unique mixtures that ranged in size from 1 to 12 components. And here we can just show that, we have the 16 individual components, and then from these individual components, we can add in other odors at the same time to build these larger and more complex mixture blends. And we started by imaging from neurons in the olfactory epithelium at the level of the nose to understand how they would respond to these complex mixtures. And if we simply just look at the population activity, so how strongly does this population of neurons respond as a function of mixture complexity? What we find is that the larger the mixture size, well, the more activity there is at the level of the neurons in the nose. And this kind of makes sense, right? As you increase the available number of sensory inputs, you're going to increase the number of neurons that can respond to those odors. And these are plotted as a cumulative distribution, but if we just look at the mean of the distributions, we can see that there's this very clear relationship between the number of odors in the mixture and the overall activity of the neurons within the nose. Now, when we tested the same idea, looking at the feedback projections from the piriform cortex into the olfactory bulb, what we found was something dramatically different. The range of responses varied quite a bit less. And in fact, the overall activity of the piriform cortex remained constant regardless of how complex the sensory mixture was. And this is especially evident when I compare this to the data that we collected from the neurons in the nose. So what this tells us is that these neurons are somehow taking the information and what's called normalizing the overall activity levels. So we wanted to understand this from the perspective of representational similarity at the level of the nose and the analytical processing which occurs in the piriform cortex. So again, we use these population vectors to compare the similarity of mixture responses as a function of mixture size. And at the level of the nose, what we found was that as we increased the size of the mixture, so here all of the mixtures that are 1, 2, 4, 8, or 12 are stacked together, what we found is that the activity of the neurons in the nose became more correlated and more similar as the size of the mixture increased. And this is somewhat unsurprising because this is what we call a 16 choose n problem. Whereas n increases, you increase the probability of sharing some of the other components in the mixture with any other randomly selected odor mixture. However, this patterning or this structure completely falls apart in the piriform cortex. And this means that our analytical processing centers at the highest levels of the olfactory system are fundamentally and rather aggressively reformatting how odor blends are encoded, even those that share a large degree of similarity. So to summarize, this portion of the talk, thus far we've seen that these neural activity patterns scale with the complexity of a stimulus at the level of input, yet remain flat in sensory processing areas. and that the olfactory cortex plays an important role in reformatting how sensory mixtures are encoded. Now, you might be wondering, what this all has to do with, feeding behaviors or metabolism. And, the answer is that, these are early days, and this is just the base. We've now been able to establish some fundamental properties of how odors are encoded at the level of the piriform cortex, where these olfactory scenes are encoded. And what we saw is that, there isn't much variation between odor blends. However, thus far in our experiments, our animals were naive to the odors. And now what we hope to do is to address whether learning about a specific odor component, like say the lipid or the sugar content of food substances, changes how these odor blends are represented in the brain. And furthermore, we're hoping to explore how reward or negative outcomes change the neural encoding of these very same stimuli. So right now, we're working on training mice to identify target odors that we're embedding in complex odor mixtures. So in this cartoon here, each of these square boxes represents one of the 16 odors in our panel. And we train mice to identify one of those odors as a target, meaning he'll get a reward if he smells that odor. So what the mouse has to do is sit and we blow an odor in his face. And if his target odor is blown in his face, he gets a sugary reward like chocolate milk. And if a different odor is blown in his face, he has to wait and not lick for a reward. And what we try to do now is trick the mice. We build different blends and distract the mice from the target odor. So we put distractor odors in at the same time and say, if your target is there, look for some chocolate milk. Or if the target's missing, don't look for some chocolate milk. And what we hope to do is combine this behavioral approach with our imaging of these piriform neurons and look at how the representational similarity changes as the mouse is rewarded for identifying specific components of olfactory blends, as well as the reward state. So the question that we hope to address, with this study is, does learning and association reformat how these odorant mixtures are represented in these cortical bulbar circuits, where context and flexible encoding strongly influence how we perceive stimuli? All right, so that sort of brings me to the last bit of the talk here, where I want to speak a little bit about some future directions that are perhaps more relevant to all of you in endocrinology. And that's our ideas for understanding modulation of sensory scenes by nutrient intake. And, I hope to expand a little bit further on these plans and some work that Vaibhav, a postdoc who'll be joining my lab, will undertake hopefully within the next year. So let's come back to this image that I showed you earlier. And let's say you arrive at Thanksgiving dinner with your extended family, and it smells and then tastes just as delicious as this picture makes it out to be. The act of eating this meal is incredibly rewarding, and our senses corroborate that fact. However, let's say that you need to go visit now with a second subset of your family later in the evening. And after eating this meal, you're confronted with the very same meal again. And this time, however, the smell of the Thanksgiving dinner is not associated with positive feelings. And in fact, the same smells that were once quite enticing may now be met with revolt. And as all of you know, for some people, this isn't necessarily the case. The second meal looks and smells just as delicious as the first. And it's this failure to dampen or adapt our senses, which is a major contributor to overconsumption behaviors. And we plan to study how metabolic and hormonal state influence how we perceive olfactory scenes, as well as their reward processing within olfactory centers. So earlier in the talk, I showed you this diagram, and I want to now highlight a structure called the olfactory tubercle. And this structure is richly interconnected with reward and arousal areas of the brain, and also has bidirectional connectivity with the hypothalamus, which is a neural area which controls both hormone production as well as metabolism. So here I want to highlight, using this diagram, the dense and somewhat overwhelming, connectivity that the olfactory tubercle shares with dozens of brain areas. But what I want to do is highlight a few of those, and those are sensory areas, arousal areas, and the hypothalamus. And if we think about the perception of a stimulus, and especially food, it's strongly connected with all of these neural process areas. We have sensory input, we have how aroused we are or rewarded we are for that, as well as our state of energy balance. Conveniently, the olfactory tubercle really sits at the interface of all of these different functions. And I think it makes it, a really unique opportunity to understand how these different neural processes come together to modulate sensory processing of nutritive uptake. So our aim is to investigate, the encoding of sensory stimuli across brain states. And our hypothesis is that the reward encoding within the olfactory tubercle is modulated by energy balance. So our approach is to implant what's called a grin lens deep into the brain of a mouse. And what you can see is that the olfactory tubercle sort of sits at the ventral surface. So we can't use traditional microscopy approaches to access these neurons. What we can do is take this long lens, which is like a tube, and the tube has an image which forms at the top of it of the neurons deep in the brain, which we then can image. And the plan is to then measure the neural activity of these dopaminergic neurons in the tubercle in both hungry and fed mice as they encounter food substances and olfactory cues that are associated with these food substances. Then we plan to manipulate the animal's internal state and energy balance through manipulations of both blood glucose, as well as using exogenous hormones. Then here's just a zoomed in of what this looks like, and you can see the image of these tubercle neurons forming at the top of the lens. And importantly, in our approach, these mice can be head fixed underneath a microscope, or they can be freely moving with a mini microscope attached to their head, so that we can get accurate readouts of naturalistic behaviors. And this allows us to capture longitudinal data, which follows the activity of the same cells in the same animals through these different states, which we can impose on them. So using the same fluorescent indicators that I earlier discussed, this GCAMP, we can track the activity of these neurons as we deliver odors to the animals. And in this case, we can deliver an odor that has been associated with an odor which has been added to their chow. And what we can see is that each time the odor is delivered, the dopamine neurons spike their activity. And what I forgot to mention is that this is a fasted mouse. He's hungry and he's looking for food to eat as this odor is being blown in his face. And if we zoom in a little bit closer, you can see these clear peaks, which are associated with delivery of the food-associated odor. Now, this mouse was hungry. So what happens if we perform the same experiment after the mouse has been fed? Well, what we see is that the dopamine neurons are much less effective at tracking the same stimulus, which indicates that perhaps the rewarding value of food-associated odor has diminished through these different brain states. So a few of the questions and goals that we hope to tackle using this approach is understanding, how reward encoding in the olfactory tubercle follows as a function of blood glucose. Can reward encoding be modulated or manipulated through blood glucose? And what hormones or neural inputs to the olfactory tubercle control reward encoding? And just to sort of sketch this out, one of the fundamental predictions that we can make is that if the blood glucose is high in or blood glucose is low in a fasted mouse, the reward encoding of these neurons in the tubercle is elevated. However, as the blood glucose increases and the mouse is fed, we expect the reward encoding to be diminished, indicating that the mouse is no longer interested or aroused in seeking these same nutritive substances as it once was. Furthermore, we want to understand, can food intake actually be modulated by manipulating the activity of these neurons in the olfactory tubercle? We can use neural activity manipulators, including both optogenetics and chemogenetics, to control the reward encoding. So rather than modulating the blood glucose, what we can do is either increase or decrease the activity of the neurons in the tubercle while holding the blood glucose constant, and measure the amount of food intake that the animal, the behavior of the food intake as the animal's reward encoding is manipulated in real time. So what we predict is if we decrease the reward encoding in the olfactory tubercle, the food intake is going to be low. And as we increase the reward encoding, the food intake increases. So to come back to the mission statement of the NIDDK, we think this work will directly contribute to this mission by uncovering how brain interprets, the nutritional content of food through sensory cues, and how that interpretation is modulated by brain state and context. We hope to uncover how sensory cues associated with food intake are interpreted by the brain, how learning changes the formatting of sensory information, and then lastly, how energy balance and satiety control the reward encoding of these same sensory inputs to the brain. And then the long-term goal for our lab is then to uncover how these processes are affected by maladaptive ingestive behaviors that lead to both obesity and metabolic disease. And the plan is to use some pathophysiological models, including diabetes, as well as fasting models, to control brain states which lead to different ingestive behaviors. So with that, I need to say thanks to our growing lab, a couple postdocs who have started recently with me, some rotation students, and of course Vaibhav, who I'm very excited to have join my group later this year or early in 2023. And of course, funding through now the NIDCD, who has made the early experiments possible and supported my work for